Welcome to this guide on navigating the metagenomic analysis of biofilm microbiome module on the NIGMS Sandbox. My name is Sonalika and today I'll walk you through how to access and utilize this module effectively. But before I do that, let us first of all understand what is metagenomic analysis of biofilm microbiomes all about. This involves studying the genetic material of microbial communities within biofilms. Biofilms are complex structures formed by microorganisms like bacteria, viruses, and fungi that stick to surfaces. This analysis is crucial because it helps us understand the composition, diversity, and functions of these microbial communities, which play significant roles in health, disease, and various environmental processes. This module, hosted on the NIGMS Sandbox, provides a comprehensive step-by-step learning experience on how to conduct metagenomic analysis of biofilm microbiomes. It's designed to help learners from undergraduate through graduate levels understand and execute biofilm microbiome analysis. Let's now take a closer look at what this module includes. The first module, or the submodule, gives a foundational overview of the concepts and the workflows involved in metagenomic analysis. The next submodule, that is on metagenome data preparation and quality control. Here you will learn how to prepare your data and ensure its quality before analysis. The third submodule will cover the analysis of the microbiome, including identifying the composition and the functions of microbial communities. Then you would learn how to discover biomarkers within the biofilm microbiome data, and the last section will teach you how to scale your workflows using Google Batch, an essential skill for handling large data sets. For detailed explanation of what you will learn in each submodule, please refer to the videos that are hyperlinked within this course. Now let's talk about how to get started with this module. First of all, you need to set up your environment. For this, follow the steps highlighted here to create a new user-managed notebook in Vertex AI. Ensure you enable idle shutdown to avoid unnecessary charges. In the environment drop-down menu, select Custom Container, enter the Docker container image provided and continue. Next, clone the repository. For this, open the Jupyter Lab and then open the terminal. Use this command mentioned here to clone the repository. After cloning, navigate to the directory for this project and open the necessary Jupyter notebooks to start with your analysis. For more technical details on the cloud setup and other resources, refer to the NIH Cloud Lab README. Now let's go over the software requirements. These tools are essential for completing the tasks outlined in the module. The first one is Docker. This containerizes applications and dependencies for consistent environments across various systems. Then you have the Jupyter Notebook. That's an open source web application for creating and sharing documents with live code, equations, visualizations, and narrative text. Next, you have the custom scripts. These scripts are tailored to specific tasks within the module. Next is a FastQC, that's a quality control tool for high throughput sequence data. The next software is MultiQC that aggregates results from bioinformatics analysis into a single report. Then you have Trimomatic, that's a flexible read trimming tool for Illumina NGS data. Next is Kind2, that's a platform for microbiome analysis from raw DNA sequencing data. Then you have Picros2 that predicts functional abundances based on marker gene sequences. Another software required would be Microbiome Analyst R. That's actually a package that's used for comprehensive analysis and visualization of microbiome data. Last but not the least, you have two other softwares, Google BigQuery and BlastPlus. Google BigQuery is a fully managed serverless data warehouse that enables scalable analysis over petabytes of data, and BLAST+ is a tool for comparing an individual's sequence to a database of sequences. Each dependency here will be loaded at the beginning of the module, and instructions for installing these tools are included in the requirements.yaml file available in the repository itself. Now, if you scroll down on the module page, you'll find the workflow diagram. The workflow for this module involves several steps visualized in the provided bioinformatics workflow diagrams and the cloud implementation architecture. These diagrams show how the raw data is processed through various tools and ultimately analyzed to produce meaningful insights. Then you have the greater insights of each and every submodule, followed by data and troubleshooting. This module uses data sets from published studies to cover the diversity of biofilm microbiomes. Detailed steps for data download and analysis are provided in the module. Common errors and troubleshooting tips are also included to help you navigate any issues you may encounter. To access the course submodules, you need to scroll up to the main page of the repository, click on the folders corresponding to each submodule. Clicking on a submodule file will open the detailed page for that submodule. Here you will find comprehensive instructions and code cells to execute the analysis steps. In addition to the submodule files, there are other important folders in the repository. The GitHub folder contains configuration files related to GitHub actions and workflows that automate certain tasks within the repository. The issue template folder includes templates for creating issues in the repository. These templates help standardize issue reporting for better project management. Quiz folder contains quiz files that can be used to test your understanding of the module's content, whereas the images folder stores images used in the documentation and the notebooks. And the kind2 analysis scripts folder holds the custom scripts related to kind2 analysis that are used throughout the module. This module provides a structured approach to the metagenomic analysis of biofilm microbiomes, leveraging powerful bioinformatics tools and cloud computing resources. Follow the instructions carefully, watch the linked videos for detailed guidance, and explore each submodule to gain a thorough understanding of the workflow. Thank you for watching this tutorial. Happy learning and exploring.